Before I start, I was asked to tell you a little something about OraChem. This will be short, but OraChem is in Naperville, and it started in 1996. And there are really two parts to OraChem. The first part, what it actually got started in initially, was bioconsumables, such as protein crash plates, SPE cartridges for sample preparation. The other part of OraChem, which makes up about 2/3 of the company now, is separations technology. This goes all the way from UHPLC columns to large-scale commercial simulated moving bed chromatography, and of course everything in between. So we do toll separations. We also set up and sell large-scale commercial separation units for customers. Train them how to do it, provide the materials that go into the columns, and work with them as long as necessary until they're up and running and feel comfortable. And we'll provide support work along the way. So, with that said, tonight's presentation is molecular modeling as a screening tool to separate enantiomers of chiral compounds using Polysaccharidebased chiral stationary phases for orphan drugs. Fortunately, the title also serves as the abstract. My co-authors are Ben-U Zhao and Priyanka Sharma and Dr. Anila Rosker from OraChem Technologies. And then Xiaoyu Wang and Professor Sohail Murad from the Illinois Institute of Technology and Professor Cynthia Jameson from the University of Illinois at Chicago. Okay. This work has been sponsored by a grant from the Small Business Innovation Research from the National Science Foundation. And our collaborators on this is the Illinois Institute of Technology. The pharmacological activity of optical isomers of racemic drugs often differs in the human body, since the body itself is chiral. Where one optical isomer may be beneficial, the other may be harmful or even deadly. Thus, separation of these racemic mixtures has become a priority for pharmaceutical companies. However, purification systems for the production of single enantiomer drugs can be challenging and expensive. Therefore, millions of people with so-called orphan diseases find potential medicines de-emphasized by pharma companies due to the unfavorable rates of return on some of these orphan drugs. The goals of this project are to significantly speed up the process and lower the cost involved in developing chromatographic systems for chiral separations. I'd like to give just a brief review of chirality. I know most of you are very familiar with this. But a molecule is chiral if it has a non-superimposable mirror image. The classic example that you see is with the right and the left hand. In the picture I show here, in the left hand, you see a generic representation of an amino acid. Its mirror image in the right hand, you see. Now, labeling the optical isomers, we use the R&S system, which determines the substituent priority based upon the atomic number of the atom nearest the carbon in this case. For example, if we look at the molecule in the left hand, and let's rotate it so that the hydrogen is pointed into the plane of the screen. That will bring the R group rotating 90 degrees out. Now, the R group represents the bulk of the amino acid. This could be a ring, another carbon with an aromatic ring, whatever. But generally, it's the bulkiest part of the molecule. Okay? Now, looking at that molecule, the substituent order is nitrogen before the carbon that's attached to a double bond, which is before a carbon attached to a carbon bond, which will be before the proton. So the proton's the lowest, it goes in the back. We rotate the molecule in the left hand, so now the R group goes 90 degrees, and basically it's like my arm's hydrogen and my 3 fingers represent the groups. So, looking at the molecule in the bottom right, the order would be amino, the acid, and then that carbon, or the R group. That's counterclockwise. That would be defined as S. So that's the S enantiomer or S optical isomer. Now, a few definitions. A chiral molecule, they come in pairs that are called optical isomers or enantiomers or mirror images. The terms are used interchangeably. A racemate is an equal mixture of enantiomers. Chirality is a three-dimensional effect. Thus, chiral recognition requires at least 3 points of interaction. And we'll look at that in more detail on a subsequent slide. Now, in chromatography, chiral discrimination is the ability of a chiral host to separate the two enantiomers of a mixture. Now, the properties of optical isomers. Well, they have the same molecular weight, the same shape, the same structure, connectivity among all the atoms, same melting point, density, solubility, energy, same everything. So when are they not the same? Well, when they're in the human body. Remember, the human body is chiral. When they're in animals or plants, or an HPLC chiral stationary phase called a CSP that's been prepared using a single optical isomer. And if we look in the lower left, we see a depiction of a chiral column, and this only has the R isomer in it. Let's say we take a mixture of optical isomers, R and S, and we place it on the top of the column and elute it. Looking at the energy of interaction between the R column and the racemate, if the RR interaction, that is between the R's of the column and the R of the racemate, is equal to the RS interaction, then we have no chiral discrimination. However, if the RR interaction is different than the RS interaction, then we can see chiral discrimination. This is the type of interaction that we'll be looking at with the molecular modeling when Ben Wu gets up and gives that part of the presentation. Now, there's a strong driving force for purifying optical isomers. And I have just 4 examples here. There are many examples in the market. Ibuprofen, for example, the S isomer is an anti-inflammatory. The R isomer has almost no effect. So basically, you're wasting half your material. If you can racemize that, then do another separation and recover the S isomer from that, you can see you can ultimately get all your product. Otherwise, 50% is lost. Looking at naproxen, the R isomer is used for arthritis pain. However, the S isomer is a teratogenic compound. Looking at thalidomide, now this is a classic example of what can happen. In the late 1950s, this was a drug that was prescribed for pregnant women to combat morning sickness. It's now used for treating certain cancers. What they found was that the S isomer caused horrendous birth defects. The US FDA prevented this compound from being sold in the US, so give them a cheer. So, citalopram, the S isomer is an antidepressant, the R isomer is inactive. So again, we've lost half the material. And like I said, there are many examples in the literature that address this. Now, polysaccharide-based chiral stationary phases. These are among the most universal of all the chiral stationary phases out there. That's why we've selected these to work with. One of the reasons it's so universal is that it has a polymeric structure with a molecular weight of over 16,000. And that offers many possible interaction sites. It's physically coated or covalently bound to, say, a silica gel particle to form a matrix that has, depth to it. It's not a monolayer. Now, the problem is they're difficult to model because it offers many possible interaction sites. Many research groups have been working on methods to predict the best chiral stationary phase and the conditions necessary for separating optical isomers. This involves the generation of very large databases, artificial intelligence, and various types of molecular models. If we look at the polysaccharide-based chiral stationary phases, generally there are 2 types. One's based on amylose, and one is based on cellulose. If you look, say, at amylose, you see those R groups. Naturally occurring, those are OHs. Amylose by itself and also cellulose do show a very low level of chiral discrimination, but it's so low it makes it impractical for use. However, if you can derivatize those, replace those R groups with some sort of derivative, you can drastically magnify the chiral discrimination capability. And that's what we've done. At OraChem, we have 5 different chiral phases that we've designed that we feel represent the vast majority of chiral stationary phases that can separate the vast majority of the racemates out there. Like I said, those that can be separated using polysaccharide-based systems. In this case, we have 2 amylose chiral stationary phases. What we call CSP1A has been derivatized with a 3,5-dimethylphenyl carbamate group that you see to the left. The material under it, 1K, has been derivatized with the 3-chloro-4-methylphenyl carbamate group. Similarly, we've taken these same derivatives and applied them to the cellulose analog. Again, 1C is derivatized with 3,5-dimethylphenyl carbamate, and 1Z with the 3-chloro-4-methylphenyl carbamate. Even though they're very similar in structure, they have drastically different capabilities for separating optical isomers, remembering that even very small changes in the chiral stationary phase can have a dramatic effect on which racemates they're capable of separating. We have a 5th system that's also based on cellulose, but the derivative used for this is 4-methyl benzoate. Again, this offers dramatically different characteristics for separating racemates. Now, if we take a small molecule, 1-phenylethanol, it's down there at the bottom, as it approaches the chiral stationary phase, come on. Unfortunately, it sees nothing. Had it really shown up, you would see 3 long chains of chiral stationary phase, which has we have like 12 units in 3 chains. So it represents a very small amount of the chiral stationary phase. However, it represents hundreds of chiral sites, all capable of interacting with that one little phenylethanol down there. So that's why it's hard to a priori tell what the separation is going to look like, whether it'll be effective. I mentioned earlier that the separation takes place using 3 points of interaction. Typically, this has been described as the 3-point interaction model and is typically observed for polysaccharides, virtually all brush-type chiral stationary phases. If we take an optical isomer, in this case the one at the top, we have a pi base, maybe an aromatic ring, some acidic group. It could be a proton next to something that draws electrons away, and a basic group. The mirror image is shown below, and all I've done in the mirror image is rotate it so that I line up the pi base and the basic group so that it matches the enantiomer at the top, but they are non-superimposable. Now I bring in a chiral stationary phase. It also has a pi acid, a basic group, and an acidic group. Looking at the interaction with the top optical isomer, we have a perfect interaction with the pi base and the pi acid, interaction with the acidic group, with the basic group of the chiral stationary phase, and likewise the basic group of the enantiomer with the acidic group of the chiral stationary phase. Bring in exactly the same chiral stationary phase as above, and now we look at the interaction with the optical isomer. The pi base interacts with the pi acid, no problem. Basic group of the enantiomer interacts with the acidic group of the chiral stationary phase. Again, no problem. But now we have a problem. The acidic group in this optical isomer is directed away from the chiral stationary phase. In its place is that R group that is bulky. Obviously, this is not going to be a good interaction. This optical isomer cannot approach the chiral stationary phase as close. Therefore, it's going to be less retained on the chiral column, whereas the enantiomer on the top, being tighter bound, is going to be retained, and that's how we get the separation. Now, selecting the best chiral stationary phase. The general procedure to do this is first check the literature. If you find your separation in the literature, great. You're extremely lucky, and you only have to do a little fine-tuning. If you're like the rest of us in the world, you wind up checking for similar structures. But you have to remember there's no guarantee of similar behavior on a chiral stationary phase. Very small changes in that optical isomer can have a drastic effect on how it interacts with the chiral stationary phase. So you're down to the best estimate, also called the educated guess. But there's no real, the real CSP racemate interaction is uncertain because, again, the CSP contains many chiral sites. It's very difficult to predict whether it's going to work. So that brings us to, well, let's try different chiral stationary phases and different mobile phases using the good old trial and error column screening method. That is the method that's currently used by virtually everybody. Why? Because it's the only thing that really works well currently. Otherwise, we wouldn't be here if we didn't think we had a better idea. Now, I mentioned the importance of the chiral stationary phase, but sometimes people don't take into account the mobile phase and the effect it can have. Even though the mobile phase is not chiral, it can have a drastic effect upon the chiral separation. In this example, we're looking at flavanone. This has been separated on a CSP 1A column, 5 micron. This is your typical column, 4.6 millimeter ID by 250 millimeter long, and using a standard HPLC. Flow rate is 1 mil a minute. The separation value is 1.06. This is really small. If we replace the heptane IPA with pure acetonitrile, exact same column, now we've increased the separation value up to almost 2. Nice baseline separation. All we did was change the mobile phase. Now, if we replace acetonitrile with just methanol, we've increased the separation value up to 2.5. But also, we've drastically increased the resolution value. This is critical. The resolution value allows us to do a large scale-up. This would be a very easy scale-up for us at ORAChem. Now, in selecting a chiral system, there are a number of factors to consider. For example, we've already mentioned the type of chiral stationary phase. We have 5 different types of coded phases that we look at. We also have 3 types of covalently bound phases that we look at. So that's a total of 8. Choice of mobile phase. Typically, we'll look at 2 normal phases, and that's really a mixture of an aliphatic hydrocarbon and alcohol, such as IPA or ethanol. So we look at 2 of those. Polar phases. That could be acetonitrile, methanol, ethanol, or blends of alcohols. And we look at 4 of those. Reverse phase. 3 different aqueous systems. We would typically look at that. And of course, modifiers, depending upon if your racemate's an acid or a base, you might need to modify the mobile phase to sharpen the peaks, or you might need pH control. So we might need a buffer in there. We'll just say that that gives 1 additional possibility. So we have 8 possibilities for the chiral phase, 10 possibilities for the mobile phase, 80 possibilities for each racemate that we want to look at. Well, if it takes 10 minutes per each run, this would take more than 13 hours to carry out this examination for 1 racemate. Well, there have been a lot of efforts to decrease the time necessary to screen racemates for obvious reasons. One technique is to use gradient mobile phases. You can do a lot of things faster. You can screen from relatively nonpolar up to very polar and do it relatively quickly. Now, you'll have to come back later and run it with, say, an isocratic system probably, but this is a fast way to do it. Most people will use an automated column and mobile phase screening device that can screen maybe 4, 6, even 20 columns. Set it up, let it run overnight. Many people are going to UHPLC and using very small columns that are very efficient. So instead of taking maybe 10 to 20 minutes for a run, we could do a run in 5 minutes or even 3 minutes. At ORAChem, we use all of these techniques, and we typically will screen either 3 minutes per run or 5 minutes per run. So we can really cut the time down drastically. But still, if you have a lot of racemates to run, it's going to take a while, and we let it go overnight. The project objective is to develop a screening technology based on molecular modeling for predicting the best chiral stationary phase for separating the enantiomers of racemic or enriched orphan drugs. The requirements are that the system must be much faster and significantly less expensive than the current laboratory column screening methods. Likewise, it must accurately model the experimental results of the chiral stationary phase drug interaction and the elution order of the enantiomers. And the solvent effects on the chiral separation must also be successfully modeled by the system. Now, at this time, I'd like to ask Ben Wu to come up and talk about the molecular modeling part of the project that we worked on.
Awesome. Thank you, Dr. House, for the introduction. So basically, the method that we're using here now is called molecular dynamics simulation. So what we do is we have a particle that represent each specific atoms in the system that we want to investigate. So in this case, we are going to look at a system that contains the chiral stationary phase, the orphan drug molecules, as well as the mobile phase, which is the solvent we are going to be used in the system. So a brief introduction of the molecular dynamics simulation. We have all the atoms explicitly represented in the system. We gave them an initial velocity. We have a predefined potentials between each system. And we push the atoms in the system. We updated their velocity, calculated force and acceleration, and update their positions according to the velocity changes after they collide with each other. So basically, and you continue with this, keep the temperature and pressure as constant, and calculate the physical quantities that you are interested in. And then you repeat this whole process until the system that you are investigating reach an equilibrium. And on the right-hand side, we have a representation of an actual system and how it looks like. In the middle, you have a chiral stationary phase molecule. In our case, we have a 12-mer CSP molecule in the system, and it was surrounded by the solvent molecules, which is the mobile phase in the actual experimentation. So what has been done previously on computational methods to investigate chiral separation? One of them is using a simulation with fixed CSP backbone by only allowing the side chain, which is the derivatives, along the CSP to move. And one other thing they did is the simulation lasts for 2 nanoseconds, which is relatively short if you consider the interaction between drug and CSP molecules. It would definitely last more than that, and there will be multiple interactions with the drug and CSP as well. Another computational study on this theme is using the docking method. So they look at the geometry of the CSP molecule. So basically look at the conformation of the CSP and see which cavity that the CSP has could have the drug fit in there perfectly. However, this is you're only considering a fixed conformation of the chiral stationary phase, and you are not losing the dynamic behavior and the interaction between the drug and the CSP in this case. Other study including density function theory, where people only look at one side chain, basically one derivatives along the CSP, and look at how this side chain could interact with the drug. So the method that we are using here, we try to add as less artifacts as possible into the system. We want to capture the whole behavior and the interactions between chiral stationary phase and drug, plus we have the explicit solvent in our system as well. So first, we would perform an equilibrium study by having just one 12-mer polysaccharides-based chiral stationary phase in the system, get an average structure based on our simulation. Then we look at an average structure of the drug. And the third step, we will put the CSP molecules together with the drug. and then we look at their interactions throughout our simulation. The average structure generated are from clustering analysis. Basically, when you get a trajectory from the simulation, you look at the similarities between the structure of CSPs and cluster them into different groups to generate the average structure. And Ember here is the software molecular dynamics engine that we're using, and the force field is called GAFF. All the partial charges on both the CSP and drug are calculated via quantum chemistry using Gaussian. The temperature is around 298 K, and the simulation lasts until 100 nanoseconds. So the initial configuration of CSP1A is a helical structure reported by Yamamoto on JAX in 2002. They reported the CSP has a helical left-handed helical structure in the solution. And one of our targets to see if we can validate the experimental observations, especially on the conformation of CSP, is to see whether we can have the left-handedness in our CSP model. There are a couple of drugs that we are investigating here. We are looking at naringenin, benzoin, tetralmethyl, valsartan, and flavanol. So first of all, as Dr. Howell just talked about, when you change the mobile phase in the chiral separation, by changing just the mobile phase, you can get a significantly different result on the selectivity and the resolution. And what we are thinking is the mobile phase is definitely having an effect on changing the conformation of the CSP. Therefore, we look at how CSP itself would behave in various solvents. And in this case, we are looking at the CSP1A, which is an amylose-based chiral stationary phase, and look at how it behaves differently in 9010 IPA and compare that with in methanol. So the first thing that we will notice here on these figures, we have the CSP structures, and the backbones are in these huge beads. They all have the left-handedness helical structure, which agrees well with the experimental NMR study. And we also look at the end-to-end distance of each these CSP molecules to see if the polymer gets swelled or not when you're changing from one mobile phase to another. And what we found is in methanol, the end-to-end distance of CSP1A is around 34, almost 35, and for in 9010 heptane IPA, it's 39. So the polymer itself, the conformation changes when you have your mobile phase going from methanol to 9010 heptane IPA. We'll further investigate how the solvents behave around the CSP molecules in the later slides. This is a representation of the actual interaction of the CSP. This huge wiggly molecule in the middle of the picture is our chiral stationary phase 112-mer molecule. And this smaller guy who comes close to it and get captured by it is the 2S flavanol that we are investigating. So here it's constantly forming hydrogen bonds with the derivatives of the CSP and get captured by the CSP. However, it's a dynamic behavior. It will eventually leave the big guy, as we can see in a couple of seconds. There you go. It's leaving the big guy. So this is a classic representation of how the CSP is interacting with the drug. I'm not presenting all the solvent molecules. In the actual simulation, there will be tons of solvent molecules around these two. And here I'm only presenting the interaction between the CSP and drug. So for the three-point mechanism that Dr. House described, when you have a CSP and a drug molecule, normally you will have a three-point mechanism, and two of them are contributed by the hydrogen bonding. And we would think that hydrogen bonds is really important on determining the selectivity of drugs by the CSP. And also from the last video that we shown here, when we look at the interaction between the drug and the CSP, we saw a constant hydrogen bond forming between atoms on CSP and atoms on drug. So we test one of the likely discriminating possibilities, and that is a transient hydrogen bonding formation between enantiomer and CSP. And since this hydrogen bonding is a dynamic behavior, you will have a hydrogen bonding deforming and reforming constantly throughout the entire process of the interaction. So we consider the lifetime of hydrogen bonds. It basically defines how long a pair of atoms can form a hydrogen bond. So one of the properties that we look at, as I just said, is the hydrogen bonding lifetime. A lifetime is defined as the length of time something remains present. So in our case, if a pair of atoms is forming a hydrogen bond, we count it as 1, otherwise it is 0. So throughout the entire trajectory of the simulation, you will get a time series of, for example, if it is a 10 frames trajectory, you will get a 2 pair of atoms that is either forming hydrogen bonds as 1 or 0. So the overall fraction in this frame is 0.6 because there are 6 frames out of 10 they are forming the hydrogen bonds. And there are 3 lifetimes because the hydrogen bond formed 3 times in this trajectory. You have this 1, 1, 1, and here is 1, and here's 1, 1. So the length of each lifetime is 3, 1, and 2. And the maximum lifetime is 3 here because this has the longest frames. And the average lifetime is when you take the 3 numbers, the 3 length, take an average of that, and that is 2.0. So when you look at a specific example of the interaction between CSP and drug, there will be many potential hydrogen bonded pairs in the system. And we count all of them, and by the end of it, we look at 4 properties. One is N lifetime, that signifies the number of times a pair of atoms is forming hydrogen bonds. And then the maximum lifetime is the lifetime of a hydrogen bond. And average lifetime is the average of lifetime of the hydrogen bonds. And total frames is the number of frames a hydrogen bond presents throughout the entire trajectory. We look at all the potential hydrogen bonded pairs between CSP and drug. We either calculate the maximum or the average of that. And then to see if we can correlate any of these quantities with the selectivity observed in experiments. And the lifetime of hydrogen bonds between flavanol, that is the first molecule that we study, between flavanol and the CSP correlates with the elution order in the experiments. We have calculated the maximum and average of lifetime. We take a ratio of R over S. All of these values are smaller than 1, as well as the experimental values, because it's R over S, and it is smaller than 1. That means S, the hydrogen bonds of S and the CSP last longer than R. So R stays in the system shorter, and R is the first one that comes out of the column. And that corresponds with the observation in experiments, which is actually the R comes out first. And then we further take the ratio of lifetime to capture the solvent effect. As Dr. House presented, when you change from methanol to heptane IPA, the selectivity decrease. And we are looking at R over S, and that is 1 over the selectivity. And all of these values are smaller than 1, means in each solvent conditions, we are predicting the correct elution order, and that is R comes out first. That is one thing. And the second thing is, see all the values of heptane IPA, of methanol, the green ones, are below the circle, which is 9010 heptane IPA. So the experimental values of methanol is 0.4, and experimental values of heptane IPA is 0.97. So all of the properties of hydrogen bonds that we look at correlates well with what observed in the experiments. So we capture the elution order, and then we capture the solvent effects based on the lifetime analysis of hydrogen bonds between CSP and drug. Then we take one step further. We want to know why the solvent are changing the conformation of CSPs. What we did is calculating the radial distribution functions of solvent molecules close to the backbone of the CSP. This is basically a quantities that can identify the density of the solvent molecules close to the backbone of CSP. When we look at radial distribution function of methanol, and this is the distance between methanol and the backbone of the CSP, you will have a peak here around 5 angstroms, meaning around 5 angstroms is the highest density of methanol close to the center, to the backbone of the CSP. And the red curve is above the green curve, indicating when you have 2S in the system, you are also having more solvent molecules close to the backbone of the CSP. And when you switch from methanol to heptane IPA, something interesting is happening here. Look at this curve with a huge peak at 5 angstroms. That is our IPA molecules. The peak, the value is up to 5.5, and when there is just methanol in the system, the peak value is just 0.5. This tells us when you have 9010 heptane IPA in the system, all the IPA molecules are being pressed against the CSP by the majority of the heptane, even though the IPA is the minority, only 10%, but the density is significantly higher even than the uniform methanol solvent condition. So this explains why we have a more swelled polymer conformation when you have a 9010 heptane IPA, because all those IPA molecules are being pressed against the CSP by the heptane, thus make the polymer more extended. So we further extended our chiral drug library by investigating more drugs in the library and looking at their lifetime of hydrogen bonds. We calculate all these quantities of the drugs we just introduced, naringenin, benzoin, tetromycyl, valsartan, flavanol in heptane IPA, and flavanol in methanol. And these are the experimental values, and these are values we observed from molecular dynamics simulation. On the last row, we are calculating the correlation coefficients. When the correlation coefficients is around, the highest it can go is it goes to 1, meaning you have an absolute 100% correlation between experiments and simulation. And in our case, we have a winner here that is the maximum of average lifetime that has a correlation coefficient of 0.96. It also has a 100% success rate on predicting the elution order, which means we can tell if R or S comes out first. And this is another figure that is showing the maximum of average lifetime has predict the elution order correctly, because all the outliers of other properties are in this red circle, but only this maximum of average lifetime is predicting correctly on the elution order with a 100% success rate. So a few conclusions of our study. The model of CSP maintains its helical structure that matched the NMR study. And the solvent makes the CSP swell when you switch from methanol to heptane IPA. And the IPA molecules are being pressed against the CSP, causing the CSP to swell, and which might cause a weaker separation than using just methanol. And the lifetime of hydrogen bonds between drug and CSP correlates well with experimental observations. A couple of the future works that we can do, we can investigate more properties, calculate more properties from the molecular dynamics simulation. But this lifetime of hydrogen bonding analysis is by far the best in terms of the correlation between experiments and simulation. We will expand our studies not only on CSP1A, but also on the other CSP molecules. One other thing that we can do is design new stationary phases using molecular simulation for a specific needs. So if someone comes to us with a chiral drug and we don't have any, the current CSP we have can't separate with all the mobile phases, what we can do is to design a new CSP based on molecular simulation, look at the interactions between the drug and CSP, do a quick pre-screening before we start all the synthesis method, trying to get a new CSP molecule. The acknowledgement that I'm going to make is Dr. Neil Oreskar, which is the CTO of our company, and Dr. Asha Oreskar, the CEO, and National Science Foundation SBIR grant. And thank you for your attention.